Welcome back, everyone. This is our second talk in today's session, morning session, and I welcome Professor Masaki. And this is a continuation to his yesterday's talk, and he's going to again talk tomorrow at the same time. And Professor Masaki.
Thank you very much. So I like to start, continuation. I mean that I'd like to explain today about the theoretical aspects, by explaining our proposal to solve this interesting system, protein clock system. And so the purpose of the theoretical model is twofold. One is to explain the mechanism of oscillations in each individual molecule. And the another is the synchronization of many molecules in a macroscopic system. So to do that, we made several assumptions. And those assumptions were based on definitions of some order parameters or some coarse-grained variables describing these molecules. I mean, a few thousand amino acid residues are here. So, and time scale is hours, so it is difficult to describe it from the atomic scale in Scratch. So the assumptions we made is to describe the system with these variables. X is the structural order parameter, varying between 0 and 1. And PD is the variable describing the phosphorylation level of C2 ring. And this varies from 0 to 1. I mean that all the 12 sides are phosphorylated, and D is 1, and all dephosphorylated, D becomes 0. And PC6A2 is a probability that the kth Chi-C hexamer binds Chi-A dimer on the C2 side. And this variable, C6BI8J, describes the probability that the kth Chi-C hexamer binds I Chi-B monomers and J Chi-A dimers on the C1 side. And Q here represents the state of how the nucleotide is bound on the C1 domain. And we assumed that the Chi-A binding is favored in X equal 1 state. So we described this time development by some kind of, looks like an ordinary chemical reaction kinetic equation. And that equation has the binding rate constant and unbinding rate constant. And we assume that the binding rate constant is large in X equal 1 state. And also we described the kinetics of D in the similar kinetic equation. And we assumed D increases if Chi-A binds here. If PC6A2 is large, then D increases. But if PC6A2 is near to 0, we assume that the D decreases, like this. And in a similar way that we assume that Chi-B binds more when X equals 0 and unbinds when X equals 1. And we treated Q as a stochastic variable. And when Q turns from 1 to 0 in X equals 0 state, then that X equals 0 structure is destabilized and kicked toward the X equals 1 structure. And when ATP hydrolysis takes place, the ATP changes to ADP, then we assume that Q becomes positive, and then that brings the X equals 1 structure to X equals 0 state. So we assume that the stochastic, ATP binding and ATP hydrolysis, ADP release and ATP binding or something. This cycle was represented by generating random variables in the model. And so we assumed X, the structure, is also affected by these chemical states. I mean that the structure transition usually takes place in a time scale of milliseconds or 10 milliseconds in usual allosteric transitions of the protein oligomers. So, but the other reactions takes minutes or hours, having much longer time scale. So we treated the X, dynamics as in a quasi, equilibrium state by, temporary, instantly constant, some external fields of P and D and Q. So given these P, D, Q variables as a static external field, then we can solve the X variable as like this is like an Ising spin under the influence of the external fields of P and D and Q. So the main assumptions in the model is the definitions of these parameters, D1, D2, D3, and D4. And by choosing these parameters positive here, that represents the positive and negative feedback. When D1 and D2 is positive, this implies the positive feedback induced upon the binding of chi A or chi B. The binding of chi A further increases X, and the large X favors the binding of chi A. So this the positive D1 represents the positive feedback effect. But D3, this minus D3 terms represents the negative feedback. When X equals 1 state, chi A binds more, and that increases D, but the increased D, large D, destabilized X equals 1 structure with this sign. But this increase or decrease of D is rather slow in time scale, with the time scale of hours. So this gradually develops after the structure switching. So this is a time-delayed negative feedback. And this term destabilizes the X equals 1 or X equals 0 state, depending on the how the nucleotide binds, ATP or ADP binds. So this is a stochastic variable, or destabilizing the X equals 1 or X equals 0 state. So this is the mechanism how the oscillations takes place in each individual calcium molecule. So if the positive feedback is strong, the X equals 1 state continues, or X equals 0 state continues. And so, if the ATP hydrolysis takes place, we can see the infrequent stochastic jump between these states. But because we have negative feedback, this jumping can be more frequent. So this gives an oscillation of individual calcium molecule. If positive feedback is weak and negative feedback is so strong, we have some kind of non-oscillatory stable state at the intermediate X. So the oscillation frequency is determined by the balance between the positive and negative feedback in this model case. And this is the constraint for the constant chi A dimer concentration and constant chi B dimer concentration. And this consists of the free unbound chi A dimer concentration, and this is the concentration of the Ka dimer bound on the C2 side, and this is the contribution from the Ka binding on the C1 side. So the total sum should be constant in experiment. And the competition between these two terms, the last two terms, induces the communication among different Ka-C hexamers. That makes the synchronization. So with this model, we saw, well, this is the simulations for the n equals 1,000 system. 1,000 Ka-C hexamers are in the simulation model. And this is one example Ka-C hexamer picked up from this ensemble. And this shows the orange here is the structural switching between x equal 1 and x equal 0. Because of the positive feedback, x equal 1 state or x equal 0 state is stable. But these cyan spikes are the ATP hydrolysis that triggers the transition between x equal 1 and x equal 0 state. And phosphorylation level follows gradually with the slow kinetics. So after the switching from 0 to 1 state, the D becomes slowly increase. And after the structure changes from x equal 1 to x equal 0 state, then that the D begins to decrease. And this is rather a noisy stochastic switching, but if we average this for over 1,000 molecules, we have rather smooth oscillations as observed in the experiment. So the experiment usually measures the oscillation of the phosphorylation level, black curve of D. So this is okay that this explains the ensemble simulation and also predicts the single molecule oscillation. And we saw that the key assumption here is the assumptions of the multiple feedbacks inside the molecule. So positive and negative feedback and kicking stochastic destabilization. And this is the case to change the these variables representing the feedback strengths, which means that this is a coupling strength between the structure and the reaction change. So if, for this case, if D1 and D2 are kept constant and D3 and D4 are changed with this scaling variable Sn, then this looks how the ensemble level oscillation of the phosphorylation level changes. So when the negative feedback D3 and D4 is weakened, then the amplitude becomes larger and period is elongated. And when Sn is getting large, then that the oscillation amplitude becomes smaller and the period becomes shorter. So with the smaller negative feedback, the oscillation is elongated and the amplitude becomes big. So this is the rather natural to considering this scheme with the emphasizing the positive feedback effect. The system wants to stay either the x equal 1 or x equal 0 state for a long time. So this is the elongation of the period. And oppositely, that if we keep the negative feedback coupling constant, coupling parameters constant, and changing the D1 and D2 by scaling them to SPD1 and SPD2, and if the positive feedback becomes larger, the oscillation amplitude becomes larger and the period is elongated. And if the SP is smaller, that the amplitude is small and the period is short. So we have the opposite tendency, opposite effect to change the amplitude and period. This is more systematic plot for changing the negative feedback strength. And this real line is a period, and the dashed line is the amplitude. So the amplitude suddenly disappears when the negative feedback is too weak, because the system is sticking to the either equal x equal 1 or x equal 0 state and does not show oscillations when negative feedback is too weak. And so interestingly, that the period is changing more than 10 times with changing Sn here. And oppositely, that if we change SP, by increasing SP, that the period increases. And here the amplitude disappears in this SP here. So this design or modification of the structural reaction coupling should bring about the drastic change of the period. Is there consequence or the prediction of the model by assuming that the oscillations are driven by the structural reaction feedback mechanism? Actually, that effects of binding of chi-A or chi-B is rather affects the entire chi-C hexamer structure. I mean, when chi-A binds this side, this proves 6 tails of chi-A subunits, and so those chi-A pulled subunits should change should be deformed in a global scale. And similarly, that if 6 chi-B monomers bind on the C1 side, each C1 subunit should somehow deformed or rotate. So the binding of chi-B and chi-A should generate such global structure change in chi-C. But phosphorylation or dephosphorylation or ATP hydrolysis in the C1 domain is a local change of the electric charge distribution. That should affect the entire chi-C structure through the communication of through the hydrogen bonding or the steric repulsion between atoms. So this should be sensitive to the single amino acid residue substitution. So our assumption is that this case, without changing D1 or D2, but changing D3 or ND4, should correspond to the case of the single amino acid residue mutation at the C1, C2 domain interface. So we expect this could explain the experimental observation that the single amino acid residue substitution changes the period drastically 10 times through the change in the negative feedback strength. Then that we can expect the similar things in the thermal fluctuation effects. So this is our hypothesis. So thermal fluctuations at the intermediate residues should also attenuate, should weaken the structural reaction coupling as similar to the substitution to the smaller volume residue at the C1, C2 interface. With that assumption, I mean, here that D3 and D4, the coupling strength of the negative feedback is weakened with this factor. With here delta EF is 5 kBT 0, or T naught is here the 30 degrees Celsius, the experimental temperature. And so delta EF is there some activation energy for inducing the structural fluctuation. So with this rather moderate enhancement of the fluctuation that for the structural reaction coupling, then that it explains the temperature compensation. Here, that the all other rate constants were assumed as a simple assumption that all other rate constants has activation entropy of 10 kBT. So without this, thermal fluctuation effect, everything is rescaled with this 10 kBT activation energy. So this is almost similar to the change in the time scale. With this, 10 kBT activation entropy, without this assumption, the system shows the period like this. But with this assumption, the system shows temperature compensation. I mean that accelerated oscillations are compensated by the weakening of the negative feedback coupling. That elongates the period to compensate the accelerated oscillation tendency. And this is the plot for changing the delta EF activation energy for the structure fluctuation from 0 to 369 kBT. So this clearly shows this is period and this is amplitude. Amplitude disappears like this in a steep curvature. This looks like the oscillations seems to be the Hopf bifurcation as a parameter of temperature. But anyway, so this is still hypothesis, but I hope that this can be confirmed through the experimental check on whether that structure fluctuation really increases in this way that weakens the structure reaction coupling. I mean that can be realized through the NMR spectroscopy measurement or some other spectroscopic measurement would be able to check this prediction. Here, this dash line. So this dash line is this curve. So this is a curve of the change of the rhythm, assuming that everything is changed with the activation entropy of 10 kBT without this assumption. So okay. So one thing, interesting thing remaining is what is the role of the ATP hydrolysis in the C1 domain? C1 domain, each subunit of C1 consumes about 10 ATP molecules in a day, in cycle. This is very slow ATPase, but we do not know the purpose or how the ATP or the free energy released by these reactions are used in these oscillations. So it should be interesting to quantitatively examine that problem. So this is the simulation results, reducing the hydrolysis frequency. So if frequency of hydrolysis in C1 domain is reduced in our model, so oscillation dies out. This is the averaged ensemble averaged oscillations, and this is the single molecule oscillations. 5 arbitrarily picked up molecules are superposed here, and we show that each individual molecule is still oscillating, but the phase are becoming random, desynchronized. That results in the almost zero average ensemble oscillation. So with the smaller frequency of ATP hydrolysis, the system is desynchronized. So this is the ATP hydrolysis frequency, and the amplitude becomes sharply drops, and period is extended like this. This is another plot. So by changing the ATP hydrolysis frequency, this is the plot for the number of ATP molecules consumed in each period. And this y-axis is the dispersion of the oscillation level defined like this. How, desynchronized. The larger d, this axis, delta d, we have the desynchronized system, so that the average amplitude disappears with opposite trend. And this numerical simulated data can be fitted by with this hyperbolic relation. So the consumed ATP number multiplied by the dispersion is constant. So if we assume the each ATP consumption release the free energy of 20 kBT, for example, it assumes that dissipation of free energy or entropy production in each cycle multiplied by the dispersion of the oscillations is about 10 kb. So what does this mean is the next issue we are considering. So this project is rather still ongoing, unpublished results, but I may talk a little about it to, and I'm happy to hear your comments about this. And things we are considering is the relation to the thermodynamic uncertainty relation, TUR. So this is the explanation I took from this literature. Udo Seifert is the person who first proposed this uncertainty relation. And this is a rather pedagogical explanation of this relation. So he considered one-dimensional system, one-dimensional lattice, and without boundary. I mean that this continues infinitely. And we considered some biased random walk along the lattice with k plus and k minus. These are hopping weights. And the difference, the ratio of k plus and k minus should be related to the free energy difference. So free energy supply should give rise to this imbalance. And this process can be represented by this master equation. So here the Pn plus n minus t is the probability that at time t, the n plus hopping toward right and n minus hoppings toward left have taken place. So that probability can be represented by this kind of master equation. And this equation can be solved exactly. The solution is the Poisson process, like a simple burst process. And from this Poisson process, we can calculate the mean and dispersion like this. So with this, for example, that the average position of the particle is like this, k plus k minus multiplied by t, and average dispersion of the position is like this. Here, the large k is the diffusion constant. So this is okay, the simple story. And we can calculate the entropy production weight like this, j plus minus j minus multiplied by this factor. I mean, this j plus, this ratio represents the free energy supply or free energy dissipation, and this factor represents the probability per unit time. So this gives the entropy production, and with this case, this can be written like this: Js here is the k plus n, k plus minus k minus, and A is the free energy difference, I mean, for the forward and backward processes. And we can define the degree of uncertainty as a ratio between dispersion and average square. And this can be also calculated because we have the explicit solution for dispersion and average. We can write like this in this one-dimensional lattice system. So when we define the entropy production during time t, this is sigma t, c. So c multiplied by epsilon square is like this. And by putting these values, we can represent c epsilon square like this. And this can be written like kb a cotangent hyperbolic 2 over a over 2. And this has a natural inequality. This should be larger than 2 kb. So this is the simple example of the thermodynamic uncertainty relation. So if we want to make epsilon smaller, to make dispersion smaller, then that C must be larger than the value required by this inequality. So that the more free energy should be supplied to make the system precise the system behavior. And this is the case for the periodic boundary condition. If we consider the periodic boundary condition, then the situation should be the same. And this is the case for the foresight. Only the foresight is illustrated here, but we can consider any number of sites. But anyway, this can represent the molecular motor cycles or the oscillation system cycles. So it has some relevance to our chi-ABC system. So in this case, dispersion of the frequency is the dispersion written by the dispersion of the period like this. So epsilon square, we considered in the previous slide, can be written like this. So delta tau over tau square. And so if we consider the time during this average period, then TUR becomes sigma multiplied by this Fanon factor. This is the dimensionful quantity, but this gives a reasonable index for the Poisson process. But anyway, this should be larger than 2 kb. So this gives, for more precise oscillations, the larger free energy supply is necessary. Then the simple question is how we can apply this inequality to our system. So some people are trying to do that, and they found some discrepancies. That the ATP consumed in the ATP chi-ABC system is much larger amount of ATP is consumed than required by this limit. But I think that the important thing is that the chi-ABC system, our system is a many-body system, not a single degree of freedom system. So I believe that the entropy production in the many body system is beginning to be, to require to become interested in. I mean, as in the recent PRA paper, we decomposed sigma entropy production rate as a sum of entropy production rate of individual oscillators. This decomposition is not straightforward, but if we consider the very short time scale that the correlated multiple change of oscillations would be rare, so we can write in this way as a sum of the individual contributions. And if this diffusion constant part is smaller, that the coherent motion is larger than the diffusive motion, then this part can be approximated like this, J square over K. And this is the part representing the many-body effect. So this J, so this is probably current or the driving force for the oscillations, is becomes stronger when phi I is behind some average phi. This is the assumption of the to approximate the oscillations with sine and cosine. And if the phase, oscillation phase of ith oscillator is behind the average phi, then the current should become stronger. And if phi I exceeds the average, then the current should become weaker. So this gives the many body effects to synchronize the many oscillators. With this representation, that the entropy production can be written like this, just by inserting this here. And the amplitude of oscillation can be written like this. If individual oscillators have the same phase as the average, then R equals 1. But if this is random, R equals 0. So this is an order parameter representing the amplitude of oscillation. So if desynchronization is not so strong, if the system is almost synchronized, so this delta phi is a small quantity, then that R can be expanded like this. So which means delta phi square, the dispersion of delta phi is represented by the amplitude like this. So if this varies when R is near to 1. And this term is the delta phi square average. And if individual D is like this, so delta D square is proportional to the delta phi square. So this amplitude can be written in the form of the dispersion of the oscillation level. So, but by putting these things, we have this expression. Entropy production rate is written with the J0 and J1 and oscillation amplitude. And if we write J1 equal some factor alpha multiplied by J0, then that entropy production rate is written with alpha and R like this. And if the system is totally desynchronized, alpha and R should be 0. I mean, if there is no oscillator coupling between different oscillators, alpha and R should be 0. In this case, we have the ordinary TUR. So the change from this should look like this. So this is an TUR extended to the case of the many-body system. Then representing R with delta D square, we have the expression like this. We substitute this equation, and this 8 or 4 pi square factor becomes 1.64 here. And this reminds us the observed relation delta S delta D square was equal to about 10 kb. So we do not know the exact way to estimate alpha yet. I believe that the alpha should be, derived from the microscopic considerations. So this is left as an open question yet. But if for the alpha is order of 1 and delta D should be less than 1, so this should be near to 10 or something like that. So we did not solve the problem completely yet, but this result already implies that to make delta D square small, about 0.1 or 0.2, delta S has to be an order of 100. That should be the reason why the 10 ATP molecules are needed to keep the system oscillate in one period. The 10 ATP molecules should be necessary from this requirement. So many things are still in open questions, but I think that this way of thinking should be, interesting, because that this is an extension of the non-equilibrium thermodynamic system toward the many-body problem. And in a biological context, so one proposal here is that this mechanism can be, ubiquitous or rather general, not except restricted to the chi-ABC case. For example, if some molecules, ATP hydrolysis triggers the structural change of some molecule, and the other molecules binds on one particular structure state of that molecule. And that leads to the depletion of the free unbound molecule. Then this should bring about the communication between the molecules. So this is the way how free energy is used to make communications between different molecules, or synchronization, or some timing adjustment can be possible through the consumption of ATP. We do not know that whether this mechanism really exists in other systems or not, but my feeling is that this is not so, could not be so specific in certain molecular systems, but it would be good to explore the possibility of this mechanism in other molecular systems. Okay, so this is the story for the chi-ABCC system, and I like to switch my talk to the other topics in later. So I am happy to have questions and.
So in general, so here is a non-equilibrium system. So the signature of this entropy production, experimentally, what will be the relation? So in the other biological context, they see it as either as a Crook fluctuation theorem or Dzerzhinsky, the work done. So here, what will be the way to prove or disprove this entropy production in the system?
So in this system, the ATP consumption, the flow of ATP is the free energy consumption, and there is no mechanical force output as in molecular motors, everything should dissipate as thermal fluctuation. So that is the free energy flow in this system. Of course, in a more complex.
So the signature of the entropy production will be calculated or
measurable. How much ATP, the rate of the ATP consumption is a measure of the entropy production rate in this case. Of course, in vivo, that the many reactions are affected each other, so that the things are not so simple. But in vitro system, that we can define the measurement.
And the temperature corresponding to that the ATP concentration production, the temperature will be the physiological temperature.
So in experiment, temperature can be tuned. So in this system, the oscillations are stable around 30 degrees Celsius. And the major oscillations can kept between the 45 and 20 degrees Celsius. And below 20 degrees Celsius or so, that oscillation rapidly dies out, showing the Hopf bifurcation. Okay, so no. So now the next topic here is the stochastic gene expression in eukaryotic cells. I'd like to talk about this because this has some favor similar to what Heiko talked this morning. I mean the diffusion and the state change, and have great overlap with what Professor Jory talked yesterday. And I think that this is a topic that the single molecule event affects the entire cell. So as Debashish told yesterday, that cell fate decision is a rather subtle naming. Of course, in the bacterial case, that the gene expression change should be directly affected in the cell behavior, but in the multicellular case, that the connection will not be so straightforward. But anyway, I like to talk about stochastic noise in the gene switching system. And this is an introductory part for the tomorrow's talk on the chromatin and genome structure and dynamics. And we know that gene expression is regulated by binding of the regulatory factor near the promoter. This is the simple case for the bacterial gene regulation. So if the repressor binds on the promoter side like this, then RNA polymerase cannot bind here. So gene expression is off. And when repressor unbinds, RNP can bind on the promoter and gene expression starts. Or oppositely, when activator binds near promoter, that recruits RNA polymerase. And if activator unbinds, the binding affinity over if the this promoter is not so strong, that RNP dissociates from the promoter and gene is switched off. So this type of the regulation constitutes the gene network, because the repressor or activators are also the products of gene expression. But from about 20 years ago, so the people are starting to measure the single cell variation of the gene expression level, and this becomes well recognized that the cell shows the large stochastic variation. This is natural because that the system is very small, and DNA is characteristically a single molecule entity. Not like a macroscopic test tube system that constitutes that includes a large number of molecules and the law of large number holds here, but does not hold here. So this is a well-known experiment in rather 20 years ago for this self-repressor case. So with the self-repression, that the product of this gene represses the expression of itself, then that expression level is narrowly distributed. But in mutants, which loses the binding affinity here, then that distribution becomes wide. The noise is enhanced without the negative feedback. So the negative feedback suppresses the expression noise. So this is a beautiful experiment, and we can see that this is a natural result expecting. But the question is it always working or so? So to see this, that we can look at the very simplified gene expression model. So when repressor binds, the proteins are synthesized with the rate G0. And when the repressor unbinds, the synthesis rate is G1. And the product protein is degraded with the rate K. And the product protein forms dimers, and this dimer works as a repressor with the binding affinity of H and unbinding constant F. And this binding rate H is a function of the amount of the protein. And an important parameter here in this simple model is the typical protein copy number. This is the G bar over K. Here, G bar is the average of G0 and G1. So this is the typical expected protein number. And the other parameter here is the adiabaticity. This is the ratio of the rate of the DNA state change over the rate of the protein number change. So this can be represented like this. F is the representative speed of the DNA state change. So the wording of the adiabaticity was coming from the condensed matter physics or physical chemistry. I mean, the optics for molecules or condensed matter. Which means that if the transition between two quantum states are very frequent, then we can consider some average state, and we can consider some excitation between these, the ground state and the average state. So this is the adiabatic case in this terminology. And but if this state change is slow, we should consider the individual two excitations. So this is non-adiabatic limit in this case. So instead of considering the quantum states, we consider the DNA states. So if the DNA state change is very rapid, we need to consider some average DNA state. But if these transitions are slower than the protein number change, I mean, this adiabaticity omega is smaller than 1, then we can consider the two different states independently. So G1 and G0 is the case like this. When the repressor binds, RNA polymerase cannot approach to the promoter to start the transcription. But when this unbinds, RNP can bind, and this starts the transcription. So this gives G1, and this gives G naught. So G naught is much smaller than G1. So this is a two-state system. And we can write the master equation like this. So this is the N is the number of the synthesized protein. And this is P is the two-dimensional vector representing two states. So this is the transition matrix. So this is somehow similar to the Run and tumble particle system as I wrote around here. Of course, that this part is different and this part is different. But you know, this is a two-state diffusion system. And so when adiabatic limit, this transition is very fast. So we can assume the quasi-equilibrium condition satisfying like this. So P1 and P naught can be represented like this. Then that program is reduced to the one-dimensional diffusion system with the effective constant for the production rate. So, you know, by this is a discretized representation, but if we describe n as a continuous variable, we can represent this with the Fokker-Planck equation or the Ramanujan equation. Then that you know, system is the Ramanujan dynamics. You know, diffusing on certain you know, free energy landscape. But in the opposite limit, the non-adiabatic limit, this part is very small. So we have the decoupled two equations for repressor unbound and bound states. So this is also the simple system we can solve. So this is the illustrative comparison of these two limits. In the non-adiabatic limit, we have the two independent states. And if Px is a distribution of the Gaussian-like distribution, of course that in a simple case, this is like a Poissonian, but you know, Poissonian is similar to Gaussian. So if Px is represented by the Gaussian functions, then that the log Px is parabola like this. So that the problem is the diffusion on the parabola surface in a repressor unbound parabola and the repressor bound parabola, and infrequently the transition occurs to change the you know, energy surface from this parabola to this parabola like this, and or vice versa. But in the opposite limit, we should consider some averaged landscape. And the problem is the diffusion along this landscape. And the difficult thing is in the intermediate case, not far from the two limits. Then we have the you know, both the diffusions and transitions like this. So this looks like an eddy or some current flux. So this eddy flux picture was proposed, but you know, this is the difficult regime to solve in an analytical way. So some perturbation theory was proposed or something like that. So this is the case. Omega, adiabaticity parameter, is near 1. So this is the case. Adiabaticity parameter is much larger than 1, and this is the case. Adiabaticity parameter is much smaller than 1. But if omega is around 1, so we have some difficulty, and we expect the noise is enhanced in this regime. So this can be seen by the numerical simulation, and the numerical simulation of this system. The standard algorithm is a GSP algorithm. Doing the GSP algorithm of this simple toy model of the gene expression, we can calculate the Fano factor. This is a plot for Fano factor with the adiabaticity parameter and the parameter for the typical protein number. And if log omega is negative, which means this is the non-adiabatic regime, we have the enhanced big noise production with some particular x. And this is the relaxation time plot. And also in this case, in the non-adiabatic regime, we have some anomaly in the relaxation time. So I think that the physics of this non-adiabatic regime should be important to understand the biological system. I mean that this experiment can be nicely explained by the adiabatic calculation. But I mean, the question is whether the non-adiabatic anomaly can be seen in real biological systems or not. And one way to describe the non-adiabatic gene expression system is the addition of the new degree of freedom. I mean, this case is an equilibrium system. The system probability flow is heading toward the free energy basin in this case, and this is a non-equilibrium system. We have the circular flux of the probability current in this non-adiabatic case. And this is the case. We have the two-state transitions for the equilibrium system and for non-equilibrium system. And this D case is our case for the gene expression system. And our proposal to explain this as a kind of continuous change of the state, by adding a new degree of freedom, we can describe this discrete jump as continuous change from this to this. And this can be done by, for example, considering the theta parameter. So cosine theta here represents the probability to stay in depressor unbound case, and this is depressor bound state. So this cosine theta should follow these equations for is in a mean field like treatment. And this is the protein concentration change. So this is the stochastic diffusion process if we add noise here. And this is the diffusion process between corresponding to the jump between two states. So this is the addition of the noise, the Langevin type equations for this coupled representations for the protein number change and the state change. And with these two-dimensional systems, we can write the corresponding Fokker-Planck equations like this. And we know that the Fokker-Planck equation is equations for the conservation of the probability or the continuation of the probability current. So Fokker-Planck equation can be written in this form of the equation of the continuity. And J here is the probability current. So this represents, in a steady state, divergence of this flux becomes zero because dT/dP is zero. But this does not mean J is zero. So there still remains the divergence-less probability current. This is the circular flux of the current. And if this, like this circular current remains here, this driving force for the system change can be written like this. This is the term proportional to the circular flux, and this is the gradient proportional to the gradient of the effective landscape, defined like this, defined like a logarithm of the steady state distribution. And this comes from the inhomogeneous diffusion or some curvature distortion in the space. And this is a plot for the case of the self-repressing one-gene toy system. We are considering the same system in a consistent way. And for this is the adiabatic case with omega equal 100, and this is a non-adiabatic case, omega equal 0.01, and this is an intermediate case. And this color shows the landscape of V. It's the logarithm of the stationary state distribution, and this is a basin, and this is a distribution projected on the repressor unbound state and repressor bound state, gene on state and gene off state. And we see the white arrows here are the circular flux of the probability current. And the probability current is almost zero around here with the compensation of the currents of the antiparallel directions. But circular flux is more evident when omega becomes smaller. And black lines here is the optimal path derived from the by minimizing the action. I mean that by representing the Langevin equation system with the path integral form, then we can define some kind of classical limit. I mean, the saddle point is the optimal path, representing the most representative pathway for the transition. It's the black line. This plot shows that by reducing adiabaticity parameter, this is log omega, the flux, the circular current increases. And as circular current increases, entropy production rate increases. Here that entropy production is rather abstract. I mean that this would not be experimentally measurable because the gene expression system is so complex. Not like an in vitro Debashish system, but this is a complex system. So, but in a theoretical way that we can define that how the system is far from the equilibrium detailed balance situation can be measured by this sigma. And this is a three-body correlation function at the different time instances like this. And so this shows the asymmetry of these three time correlation functions. So this asymmetry in three-time correlation increases as flux increases in non-adiabatic regime. And also, I'm sorry, this is the entropy production rate, sigma. And this is the dispersion, Fano factor. I'm sorry, this is a confusion of the notation. Anyway, so noise is enhanced, and non-equilibrium feature is enhanced as non-adiabaticity is becoming prominent. By departing from the adiabatic limit, non-equilibrium feature and noise feature are enhanced. So, in bacterial case, we can see that the typical time length for the degradation of the proteins is about similar to the cell cycle time scale, is a few tens minutes or something like that. And unbinding rate is, inverse of F is about a second or seconds. So this gives omega is much larger than 1. So it is natural to assume the adiabatic approximation in the bacterial case. But in higher organisms, in eukaryotes, we have many slow processes like chromatin structure change and histone modification and the DNA methylations. These time scale of these processes should be sometimes more than hours or days. So these processes should be non-adiabatic. So I would like to emphasize that the effects, non-equilibrium effects of non-adiabaticity should be prominent in eukaryotic systems. So to see this, we simulated, again, the simple system. We consider the chromatin state with having 3 states, the active state, neutral states, and repressive states. With S equal -1, so 9th lysine in the histone 3 are methylated, and that induces the aggregation of the nucleosomes like this. And here, the lysines of the histone tails are accentuated. That gives positive charges in nucleosomes, and that should give rise to some repulsion among nucleosomes to expand the chromatin structure. So that facilitates the factors necessary to transcription approach to DNA. So RNA polymerase II can enter into the structure like this, but expelled from this condensed structure. And this structure is further induced by the transcription itself. So this is a feedback mechanism. So action of the RNP2 changes the histone marks from H3K9 methylated state to H3K9 accentuated state or H3K4 methylated state. So that is because that some factors necessary for transcription can recruit histone modifier enzymes. So this is some feedback mechanism. Anyway, so we have the 3 chromatin states, and transition time scales is not so fast. Sometimes minutes or hours. Particularly when the DNA methylation is involved, it should take days or something like that. It should be longer than the time scale of the cell cycle in that case. And this part is the same as the previous bacterial model. So by incorporating this chromatin state and coupling with the simple gene expression model, again, we can solve the problem in a GSP algorithm or the method to represent the DNA state change with continuous variable by adding extra new variables representing the DNA state change and chromatin state change. So we introduce here the two additional variables. This is the case. This axis is the protein concentration, and this axis is the histone state change. So at the beginning, we assumed the three discrete states, but we connected these three states with the continuous variable in a model. And we can see the circular current around the basin of the landscape. This is the case of one gene self-activator, so that the gene product is activator. So when it binds, it recruits RNP2 and enhances transcription. And this is the case for the self-repressor. This is the case that product protein binds to DNA to repress gene expression. So this case, we have a definite circular current around this basin, and we do not see the clearly, but we have in this case two basins here in the high transcription state and the low transcription state here. So we have two basins. So we have the switching behavior between these two, and the probability current connects, run between these two basins. An interesting thing is the direction of this current, so that the nonlinearity of this system determines the current direction, and this suggests the chromatin state change takes place fast, and then the protein concentration change follows. So because that in this self-activator or self-repressor system, the protein concentration change and the histone state change is coupled, so it is not obvious to see which comes first and which comes later. But with this model, the way of the coupling brings about the definite order of the changing. So actually, that in many molecular processes during the cell differentiation from the the early timing of the development, histone state change is earlier than the gene expression change. So we have not the quantitative comparison between this, but this suggests this is possible because of the coupling through this feedback loops. The chromatin state can change earlier than the protein state change. So I mean that there has been a long-standing argument on which is the cause and which is the result, or this is like a hen and egg problem. And by solving the quantitative model, we can discuss that which comes first and which comes late. I mean, these cause and result are coupled in a feedback loop. So this is an answer for the quantitative physical view for the long-standing argument on the hen and egg problem. Anyway, so this is a correlation. The positive value shows that chromatin comes fast. Well, this is the case. DNA state change, binding and unbinding of the repressor or activator is not so fast. In that case, we can consider the three-dimensional space, and we again that the qualitative conclusions are the same. We have the two basins in the self-activator case and flux current connecting between these two basins. Okay, so this is the case for the two-gene mutual repressor, as Mohit Jory talked yesterday. So this gives the switching between two basins. We have two basins for the protein 2 concentration and protein 1 concentration. And for this parameterization, for this set of parameterization, if the chromatin state change is fast enough. This is not so realistic, but if chromatin change time scale is faster than the protein number change, then we have the single bases. But we have the circular current. And by reducing omega to a more realistic case, we have two basins. And we see that this circular current is a precursor of this basin splitting. This is Hopf bifurcation. So I think that this flux current view helps understanding of the landscape view of the gene expression is an interesting aspect. And we are now considering the more complicated, the larger gene network. But I will stop here. And tomorrow that I will talk about chromatin and genome, because I believe that this chromatin state dynamics is important to understand gene expression in eukaryotic cells. So thank you.
With this, we thank Professor Sasai for giving a very interesting talk and giving, at least to me, more insight to theoretical understanding of this synchronized oscillation and gene expression. Questions? Yeah, sure.
So Masaki, probably I have missed a point, maybe you have already mentioned. So when the polymerization is going on, so the polymerase is opening the the DNA from the chromatin, or there is chromatin remodeling enzyme which is opening?
This is of course, important problem. And the recent cryo-EM structure analysis, I mean that with cryo-EM observations, we can see the snapshots of the continuous process. And that shows RNA polymerase 2 opens the DNA and moves the positions of the nucleosome from forward to the rear. Mm-hmm. And then the remodeler modifies, corrects the nucleosome to the right position after that. Is the observation of the cryo-EM.
For bacteria or for eukaryote?
Eukaryote.
In eukaryote, I see.
I mean.
So anyway, yeah, for bacteria, of course, that would not happen because there is no chromatin. Yeah, okay.
Thank you. Any more questions?
Hello. Hi, sir. So this is more of a doubt. So there was one slide where you showed that there were two parabolas representing DNA on and off state. So I just wanted to know, maybe I missed that point, that those parabolas where they were intersecting each other. Yeah, this slide. So the more intersection they'll have, the more there will be the scope of the eddy flux. The more separated they will be, the more stable DNA on and off state will be present. Is that so?
The more transitions we have,
Okay.
I mean that the distinction between this and this would be obscured. I mean that the average should make sense.
Okay.
I mean that this position, of course, would be a function of the omega, but I did not emphasize that.
Okay.
Other questions? Okay, let's thank Professor Sasai again. And with this, also thank both speakers of this session, Professor Reger and Professor Sasai. And we are heading for the lunch.